So, welcome to OA Week 2025. We will wait a few more minutes maybe to allow people to join us. I can see attendees are dropping in. All right. So, in the name of time, we're going to start. Welcome to OA Week 2025, everybody. We're thrilled to have you here. This is the first session of OA Week 2025, so special welcome. And thank you for joining us. My name is Katarina Shaw. I'm an Associate Project Officer for Ocean Acidification at the IOC of UNESCO. And I would just like to point out that this session is being recorded. OA Week is presented by 4 sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA, the United States National Oceanic and Atmospheric Administration. Third, the IAEA OAICC, the International Atomic Energy Agency, Ocean Acidification International Coordination Center, and last but not least, the IOC, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization, the UNESCO. So GOA-ON was established in 2012 with just a handful of members, and since then it has grown immensely. It now has over 1,200 members from over 110 countries, and also has 11 regional hubs, which span across continents and oceanographic regions. We'll be hearing from most of them throughout OA Week. And if you're not a member yet, you can visit GOA-ON.org and join us. OA Week debuted in 2020 and returned in 2021 when events and conferences were postponed due to COVID. Following the successful in-person symposium on the ocean and high CO2 world in 2022, OA Week has become an annual event to maintain the momentum around ocean acidification research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We're thrilled to present a wide range of ocean acidification topics and speakers from around the world this week. I just want to add a few words on housekeeping. During the presentation, all attendees are in listen-only mode. You're welcome to type any questions you have into the Q&A box, which can be found at the bottom of your screen. We'll be monitoring incoming questions and we'll pose them to our speakers during the question and answer section, which will be at the end of this presentation. For the discussion, you can also use the raise your hand function, and we will call on you and you can ask your question directly. And with this, I would like to introduce you to our moderator for the session. Sylvain Agostini is a researcher at the Entropie research at the Institute for Sustainable Development in New Caledonia. And prior to that, he spent the last 20 years in Japan, where, together with colleagues from Japan, France, and Italy, he initiated the ICONA project. So Sylvain will introduce the project to you all in more detail. And with that, Sylvain, the floor is yours.
Thank you, Katrina. So as Katrina introduced me, so my name is Sylvain, and I try to moderate to this time session. So we have been doing this like contributing to OA Week for the last 3 years, I think. And every year we were like always going overtime. So this year we have chosen 3 speakers only to try to keep some more time for Q&A. Just before going to our main speakers today, I want to present a little bit what we are talking about. So I hope you can see my screen. So today we'll be talking about the activities, presenting some of the activities of the International CO2 Natural Analog Network. And as I said, my name is Sylvain Agostini. So I'm working now for the IRD in New Caledonia. Previously, I was in Japan at the Tsukuba University Shimoda Marine Research Center. Well, today is the first session of the OA Week, so we can still remind everyone what is oceanification, but as you know, OA is due to excess CO2 that will release in the atmosphere that being absorbed by the ocean, and this has led already to decrease in pH, and prediction goes that the pH will decrease to 7.8 in the worst scenario, which is a massive drop in pH when you think about acidity of the ocean. And the problem we have been tackled with ICONA is that you can do experiment in aquaria, but this will be mostly single species, or maybe a couple of species will be short-term experiment under fake, I would say, non-ecologically realistic conditions. And this has limiting our knowledge of the effect of oceanification at the ecosystem level, and therefore it's very difficult to predict the performance of this ecosystem, the distribution of this species under natural conditions. In 2008, there was very important paper by Jason Hall-Spencers and also Riccardo and some Italian team in Ischia, which is maybe the first natural analog of oceanification, CO2 vents being used as this. And where so they used a volcanic carbon dioxide vents to show ecosystem effects of oceanification. And this kick-started a new field of research where we use this kind of ecosystems, so two major types, volcanic CO2 vents and semi-enclosed bay. They have chemical condition at least, for example, CO2 as much as CO2 as you have at the end of the century, or sometimes that can exceed the future ocean conditions. And so these are kind of extreme environments that are and can be used and are being used as natural laboratories to understand the effect of OA and also the stresses on marine ecosystems. So in 2019, I think we started the ICONA projects through funding from JSPS in Japan. This project started with colleagues in IRD, where I'm now, in Palermo University, and other colleagues in diverse universities in Japan. Now I think like there are more than 40 members, active members at least, and we have been using sites, so from volcanic vents, as you can see in the photo where you have lots of bubbles, to semi-enclosed bay, so mostly two in Palau and in New Caledonia, the Burakes Lagoon. So vents are located in volcanic active regions, such as the Med, the Mediterranean Sea, or Japan, or Papua New Guinea, for example. We have been working quite a lot. And for the moment, we use two semi-enclosed bay, one in New Caledonia, one in Palau, the one in green here. In 2020 something, ORS endorsed ICONA as an action for the United Nations Decade of Ocean Science. And since then, we have been trying to contribute to the ORS activity too. Very rapidly, because I don't want to eat all the time for my colleagues, but shallow hydrothermal vents. So these are volcanic active systems where the magma will heat and warm up and release gas that will go through the crust and will mostly be released as streams of bubbles, rich plumes of bubbles in the water column, and this will acidify the area around. This is an example of Shikine, where I've been working a lot. And you have a main vent in the left, represented by the red triangle. But we don't work too much here, as it's too extreme. We work nearby, in a less extreme area, where you have PCO2 of about 800 ppm, but also very variables. And then we use a control bay nearby again, where you have normal 300, 400 PCO2. So to give you an idea of the ecosystem shift we have been seeing, is in the control seen in this photo, you have corals and various macroalgae. This is close to the main vent, so not so much to say, like bubbles, of course, but what is interesting is this was the topic of another talk last year at OA Week, this bacterial mat also, which contains, can contains and have hosts some, for example, cyanobacteria, to the case in Vulcano, that can capture CO2. So there's still something to learn from here, although not as a window to the future ocean. And when we move to the OA, so in a range like the area, you have lots of turf and very few corals. So this is just an example of the kind of shift and the three locations, the control that you use, the window to the future, and the more extreme environment where you have some kind of crazy metabolic activities going on and a unique ecosystem. This is an example of the Burake Lagoon. Okay, so it's a lagoon enclosed into a mangrove, but there is no rivers. And the water will flow in and out with the tide. And when it flows out, you have the water coming out from the mangrove that is deoxygenated, acidified, and warm. And so what you have is you have a very variable pH depending on the tide level, and very variable DO also. In the most extreme, the pH can decrease to 7.4 and come back to 7.9, and which compares the control with stable pH. So here you see slightly different. It's not volcanic, it's multi-stressors, and it's very variable but predictable. And both have their advantages and inconvenience. And so in the ICONA project, we have been using both. Oh, this I want to skip because I don't have time. So what we can learn from the volcanic vent is, and this semi-enclosed, is a biological effect. So we can look at the chronic exposure of marine organisms to elevated CO2. We have been working on the molecular, the physiology, or the behavior also for fish. And this has helped us to understand the effect, but also what species traits can confer resistance to expected elevated CO2. We can also look at what is more interesting and more unique even is the ecosystems effects. And we have shown that all the high CO2 condition alter mechanisms, they often go towards simplified marine ecosystems. There are still some resilient species that can still provide some ecosystem services. Hopla, there's a wrong thing, sorry. It's actually the wrong PowerPoint, that's why. Let me start again. Okay. But so in the second phase of the ICONA project, what we want to more and more develop and what we have been starting to work on is actually to use these natural analogues to develop nature-based solutions, because in these natural analogues, we have some organisms that have been exposed for their entire lifetime, sometimes for multiple generations, to the future ocean and to extreme environments. And so we could use these ones for acid evolution, acid gene flows, or even for discovery of organisms capable of capturing CO2. And the last talk of this session will be on why we should conserve these ecosystems and what is status now. But if you look at least Vulcano, because semi-enclosed bay, there are many more, I presume, but volcanoes, you have volcanoes everywhere in the world. We know from literature that there are researchers working a little bit everywhere, even the Caribbean or the west coast of the Mexico. And honestly, like, just contact us because we want to increase our collaboration network. We want to increase sites that we are working on. And I think like the more we are, the better it is, so we can replicate our studies and we can help each other. So don't hesitate to contact us. And now I would like to give the floor to our three speakers today. So we have two early career scientists, both from University of the Ryukyus, so one just moved recently. Oh, Michael Izumiyama also. And so one is more ecological side and the other one is more genomic. And to conclude this session, there will be Milazzo from the University of Palermo, talking about threat and conservation of shallow hydrothermal vents. So perhaps I don't need to take a question now. I think we can keep them for the speakers, but we'll have a Q&A at the end of the session. So don't hesitate, stack on some questions, and feel free to ask. Okay, that was all for my introduction. Let's move to our first speaker. So it's Britta Fiedler, PhD student from the University of the Ryukyus, who will be talking about potential winners of oceanification, benthic community at a CO2 vent in Papua New Guinea. So Britta, let's try to keep time. So about 15 to 20 minutes.
Okay, what a challenge. Thank you so much for the introduction, Sylvain. So hello, everyone. My name is Britta. I'm a PhD student at the University of the Ryukyus in Okinawa, Japan. And today I will present an ICONA project from Papua New Guinea, where we assess the benthic communities at a CO2 vent. So I also have a little bit of introduction on ocean acidification, so I hope I don't repeat too much. So we know that ocean warming is one severe consequence of climate change following human-induced CO2 emissions, and we've experienced the rising CO2 emissions since the Industrial Revolution in the 1800s. Now, the oceans are a major CO2 sink, absorbing about 31% of anthropogenic CO2 emissions. Now, the current global ocean pH is 8.04, and that already is an increase of almost 18% only since 1985. Now, based on the current policies and actions, the median increase in temperature is estimated to be 2.9 degrees Celsius by the year 2100, which means that the pH would drop down to 7.7. But why is it a problem? So basically, the added CO2 changes the water chemistry in the ocean. Overall, when atmospheric CO2 dissolves in seawater, it forms a series of acid-base equilibriums, effectively forming carbonic acid. The result is a higher concentration of hydrogen ions, which makes the water more acidic, but at the same time, it reduces the availability of carbonate ions, which are essential for biotic calcification. So as a result, calcifying organisms such as corals have troubles building and maintaining their calcium carbonate skeleton, as less carbonate ions in the water mean reduced calcification rates and increased erosion. Now, as ecosystem engineers, corals are of a particular concern, but they are not only losers on the reef. So the effects of ocean acidification on corals are generally well understood, but there are other reef groups which are very understudied, and some of them have shown to be more resilient to low pH. One of these groups includes sponges, which have a very simple body plan. About 92% of all species have a skeleton comprised of collagen fibers and silica spicules, and only very few have calcium carbonate, so this could explain the higher tolerance to low pH. In laboratory experiments, 17 out of 25 species showed a neutral or positive response to ocean acidification, but in nature, this tolerance varies. So on a reef with a volcanic CO2 vent and low pH in Milne Bay, Papua New Guinea, two sponge species, namely Synechaira, which you see here at the top, and Coelocatera singaporensis, were found to be 40 times more abundant at the seep, while others declined. So there seems to be a species-specific sensitivity among sponges. Now, another group known to be resilient to anthropogenic stresses are octocorals. They do have calcite spicules, but they're only small, scattered, and buried inside the tissue, which means they generally have a low calcification sensitivity. At a subtropical CO2 seep in Iwo-Torishima, Japan, researchers found a coral to soft coral gradient with decreasing pH, with the encrusting soft coral sarcophyton, which you see here at the bottom, being most abundant in a medium PCO2 environment. At high PCO2, meaning very low pH, both soft corals and hard corals were absent. So here we can also see that also soft corals are species-specific. So how can we expand our knowledge on these groups and how they will perform in the future? So Sylvain already introduced the ICONA project and the sites where we conduct research. So I'm not going to go too much into detail. So here on the map are the locations in Asia. And for the tropical ones, there's Papua New Guinea, which ICONA has been exploring since 2016. Now, for this study, the goal is to assess benthic community changes over time in Papua New Guinea. Specifically, I want to find out if there are changes between the years 2018 and 2024, and how the communities differ between different pH levels. In the photos here, you can see Ambital Island. It's a small island, part of the Fenny Island chain, located about 60 kilometers east of New Ireland Island. And here we have a year-round water temperature of around 30 degrees Celsius. Inside Tutum Bay, which is a very small bay on the west coast of Ambital Island, shown here in the photo, there are 4 big hydrothermal vents at about 8 meters of water, and hundreds of small diffuse vents where between 93 to 98% pure CO2 gas is being released. The pH inside the bay ranges from 7.62 to 8, depending on the location and the water depth. Now the pH is lowest here in the shallows in the back of the bay and increases the further you go out to the left and the right side, and also the deeper you go. The reference site is about 500 meters south of Tutum Bay and has a pH of 8.03. Now, despite the low pH, Tutum Bay has a rich benthic community with about 140 scleractinian coral species, lots of Clavularia soft coral, which you can see here in the picture, some Halimeda algae, sponges, ascidians here in the lower left corner. And often these groups grow right next to a hot vent, as you can see here in the photo. So for this study, I analyzed benthic transect data from 2 different years. Generally, the data were collected from 2 depths, from 5 meters and 8 meters, and for each depth, 6 times 10-meter transects were laid. For both years, ICONA members collected the data and kindly shared them with me. For 2018, the data were taken in June at the vent site, specifically here in the back of the bay, which we consider low pH, and at the control site. So in total, I have 120 meters of transect data for each of the sites. Then for 2024, the data were collected at 3 different vent sites inside the bay, low pH, here again back in the bay, and then the left and right sides of the bay we considered mid pH, north and south. For the control sites, we added another control a little further away from the original control. So in total, that means I have 360 meters of transect for the vent sites and 240 meters for the control sites. I used the line intercept method, identifying the benths every centimeters according to these 8 benthic groups. I mostly identified to genus level, except for sponges, which I grouped into morphological OTUs. Now, the results for this study are still very preliminary because I only got them a few days ago, and it's the first time I'm presenting them, so I'm quite excited. So far, I've only analyzed a few sites and no control. So the sites I've already analyzed include the 2018 data collected in the bay, considered low pH, and the 2024 mid pH data from the left side of the bay. Now, for the 8 and 5 meter transects, the total difference in pH was small, with 7.9 on average for 2018 and 7.95 for 2024. So looking at the pooled results for both years at the vent sites, we can see that the coral cover declined from 20% to about 15%. For encrusting algae, the decline was a lot more dramatic, decreasing from 28% to 10%. Well, at the same time, we see an increase in turf algae from almost 19% to about 30%. What we also see is an increase in sponges from 5 to almost 15%. Now, in this graph here, I split up the benthic groups by transect and depth. So we can see bigger changes in the deeper transects, which are the 2 ones here on the left side. And over the years, encrusting algae declined by 32 percent points from almost 36% to 4%. But looking at the 5 meter depth, the change wasn't as dramatic as it only decreased by about 3%. Now, the overall turf and sponge incline was consistent for both depths. So the sponges went up from 5 to 11%, for both years, so that's about a 6% increase. And turf algae increased by about 12% for both years. So the cover for octocoral, for example, was a bit surprising because while it was overall stable between 2018 and 2024 at about 11%, there is a steep incline for the deep transects here from 7% to 18.5%, but the cover actually declined for the 5-meter transects from 14% to almost 1%. So from these preliminary first results, we can see an overall decline in hard corals while turf algae increases. Now this indicates some large-scale mortality, probably bleaching induced. And since the region in Papua New Guinea is so remote and therefore understudied, there was no bleaching reported between those years. But looking at the NOAA Coral Reef Watch data, PNG has had several consecutive months of bleaching alert level 2 every year since 2020, so mortality is quite likely. The graph here on the right that you can see is an example. Here I pulled out data from 2022 to 2023, and here you can see that especially from November, December 2022 all the way up to May 2023, the region had increased heat stress for over 6 months. What we also saw in the data was a decline in encrusting algae, and as they act as a substrate for coral larvae, this could mean reduced settlement rates of corals and therefore also, unfortunately, a slow recovery in coral cover. Now, the increase in sponges is definitely something I'd like to investigate further. I have barcoded some sponge samples using the 18S gene as a marker, and after blasting against the NCBI database, most samples turn out to belong to the genera Micale and Dissidea. So both are known to overgrow corals, but they are not bioeroders. And after annual heat stress for the corals, and we also observed some diseased corals in 2024, it is quite likely that the sponges took advantage of weakened coral structures and overgrew them, as you can see here in the photo. So the darker patches are an encrusting sponge. Interestingly, Dissidea was also reported to commonly overgrow corals on Kimbe Bay in Papua New Guinea. So I would say it's quite likely that this genera is among the sponges we also find in Tutum Bay. So there's still lots and lots of work to do. And regarding my next steps, I want to finish analyzing all the sites. And I also have data for shallow transects, which were laid in about 1 to 2 meters of water. So this depth also has a lower pH compared to the 5 and 8 meter depth transects. Then secondly, I want to refine the functional groups for all benthic groups, because I started analyzing the data before I had the opportunity to go and see the site myself, which was just this year. And after discussing with the project group, we decided to add growth forms, especially for corals, as studies have shown a decline in structurally complex corals at CO2 vents, but also for algae and sponges, since we mostly saw encrusting sponges present at the seep site. Then thirdly, I would like to identify those sponges to the lowest taxonomic level possible. I mentioned that the most abundant groups are likely Dissidea and Micale, but I had a similarity of less than 97% when I blasted them, which is quite low for sponges. So still not quite sure if even the family is right. And fourthly, I would like to add some more data from this year. So in 2025, we laid more transects in the 3 event and the 2 control sites. And since last year, many corals were diseased, we want to check if the coral cover has declined over the last year. And then this year, we also collected data for a range of different projects. So for example, with the help of eDNA, I want to assess the effects of low pH on the diversity of the very overlooked and understudied rubble fauna living in dead coral framework. For this, we collected rubble at different pH sites. So here at the bottom, you can see me collect rubble, put them in buckets. Then we brought those buckets back to the boat, let them soak there for 2 hours. And then we stirred the water with the rubble and then collected the water and filtered it. Then secondly, we also saw a lot of coral recruits this year. So we measured all recruits along the transects that we laid to see if there are differences in recruitment between the vent and the control sites. Then there's also eDNA data from 2024 waiting to be processed and analyzed, as well as some fish videos to look at the fish communities. And lastly, over the past 2 years, a lot of coral transplantation experiments were conducted by Ricardo and Sylvain to test for long-term and short-term low pH exposure on the gene expression of corals. But all of these projects that I mentioned are still being analyzed. So you can see a ton of data were collected in PNG by ICONA over the last years, and the results will hopefully help us understand what coral reefs in a high CO2 future would look like. So I would like to thank the whole ICONA project team for the opportunity to collect data in PNG, as well as for help in the field, sharing data and supervision. As well, this work would not have been possible without the French Flotte Oceanographique for providing funding and logistical support on the RV Antea to visit such a remote place. So I would like to thank the crew from the PNG Explorer who supported us this year. And most importantly, I would like to thank the local landowners of Ambito Island for letting us do research in their waters. Thank you very much.
Thank you, Britta. It was great to see your results, although preliminary, but still great results. It's kind of sad to see that bleaching is even affecting PNG and other sites. I see in the chat we have some PNG participants. I mean, like coming, but yeah, it's pretty sad. Although I'm looking for the future results to see if the combination of high CO2 with heat stress change the trajectory, the recovery trajectory of coral reefs. I think it will be pretty interesting. Okay, now it's time for question. Actually, we have already a question from some anonymous, but the question is actually linked to what I said. So do you have any data showing eventually that coral mortality was higher at the CO2 vent because of the combination of OA and high temperature? Or what's your opinion?
So in Ambito, we actually have a stable temperature of 30 degrees Celsius. So we more look at the sole effect of ocean acidification on the coral community and not so much the combined effect of heat stress and ocean acidification. And at least for this year, the water temperature was between 30 degrees and 31 degrees Celsius. And I don't know about you, Sylvain, but I didn't really see coral bleaching in those waters. But at least for 2025.
Yeah, I think there was like definitely some mortality in between the different surveys. I mean, the site greatly changed, and we've seen a drastic decline of corals, as you showed, even in your data. Now we need to go more into the detail to see if there was a combined effect of pH. But by comparing, like as you did, to 2018 and 2024, we may know a little bit if the OA and high CO2 and temperature can act together in a bad way to decrease coral mortality, to increase coral mortality. We still have a few minutes if you have some questions. If not, what we can do, we'll keep some time at the end and we can come back to you, Britta, for some further questions. If there are some further questions at the end for you. If there is no question, I would like to propose that we move to our next speaker. Thank you, Britta. Oh, no, there is another question. Hi, okay, last question for you. Okay, how were the reefs otherwise? Healthy is the question. So I don't know if the reefs only in the high CO2 or all around. Maybe all around. I don't know. What do you think? They are healthy reefs or not?
I feel like with a shifting baseline, it's pretty hard to define what a healthy coral reef is nowadays, right? But at least I would say we did see even a difference between the two control sites, right? So the control site that was further away did have more living corals, and I would say a higher diversity, but I haven't analyzed my data yet to confirm that the diversity actually increased. But for sure, it did look, well, it did have a higher coral cover, I would say, and it did look better, but it was not a pristine, untouched, completely healthy reef. That's unfortunately not the
Yeah.
case.
Yeah, I agree. I think PNG, the image of PNG reefs are very pristine reefs, very healthy. We were starting to see some decline. Okay, we take a last question and then we move to Michael. Sorry, Michael. So did you see, the question is, did you see any tapir sponge or sponge that are known for modern corals? I think it's everyone's question because we talked a lot about sponges.
Yeah, so I actually worked on Terpios a little bit before for my master's thesis. So we did see some encrusting sponge, especially one of the photos that I showed earlier, which was on the diseased Marilinid coral. It might look like Terpios at a first glance, but I don't think it is because it might behave similarly, but it just looks very, very different. And I haven't had the chance to amplify and further look at those sponge samples. So I am pretty sure that it's not Terpios. Might be some Calinula sponge, but yeah, this is still to be determined.
Yes, thank you very much. Okay, there is one last question, but we need to move. But I will put a link to so the question in the chat is do you have any data on pH fluctuation around CO2 vents and the control sites? So just give you a link to a paper that was published by Pitcher and Ricardo at the same sites, and I'm pretty sure there is some here DL variations published data. But yes, it's a very interesting question. It's one of the goals of ICONA also is to mutualize our efforts to get the chemistry at each site well defined. And so when we go to a cruise, a field mission, we try to get pH data and CO2 data, etc. for everyone in the cruise. Thank you very much, Britta. It shows well that a young scientist can do good also in ICONA. And Michael, I give you the floor.
All right, thank you, Sylvain.
No
Let me go ahead and share my screen. All right. Everything look good? Cool. All right. So thanks again. So my name is Michael Izumiyama. I'm a postdoc at the University of Ryukyus. But I'll be talking about some of the work that I actually did during my PhD at the Okinawa Institute of Science and Technology at the Marine Climate Change Unit. There we go. Cool. So as we've been talking about, we're all here to study the effects of climate change, especially OA. And you might be wondering, well, how do you study these things? How do you study the effects of OA? And a lot of great foundational work has been done with aquarium studies, where you look at controlled environments and you pump CO2 into it, and you look at the effects of it. But again, as Sylvain kind of mentioned in the introduction slides, it's not a realistic scenario in terms of a natural ecosystem. It's also a lot of hard work. As I'm finding out now, I'm actually doing some tank experiments as I'm working on things, and it's a lot of intense work, and it's really hard to do long-term experiments or look at multiple effects of environmental connectivity and how ecosystems react to the shift. So what you can do is you can actually look at this in a natural environment. And this is where some people have actually created elaborate systems where they pump CO2 into, they make a flume and pump CO2 into the system and actually expose some corals to conditions of OA and study the effects of this. This is really interesting and cool, but that's also a lot of work to do it. And that's where these CO2 seeps come into play. You already have a volcano that's pumping CO2 into the surrounding environment, and you can study the effects of OA. And this is all kind of the introduction that Sylvain's kind of talked about today. And this is one of the cool sites that I actually got to work on for my work is at Shikine Island. So Shikine Island is a volcanic CO2 seep located off the Izu Peninsula, and it's actually part of Tokyo. So this is part of Tokyo Prefecture. And it's actually a really beautiful island where there's a lot of hot springs all around the island. They're all free to use, and you can come in, and it's probably one of the great experiences of my research when I was doing my PhD on Shikine, is after long nights of diving and collecting samples, we just get out of our wetsuits, jump right into these hot springs, and you get to warm up and enjoy, just kind of unwind before you get back to the lab to do more sample processing and things like that. And here's some videos that we actually took at the seep. So on your left, you can see the CO2 seep where bubbles are coming out of the vent. And then to your right is the control reef, which is only about 500 meters apart. And you can see that there's actually a difference in the habitat structure. You see a lot of turf algae in the seep versus coral reefs. But in both sites, there's actually a lot of fish found in the two sites. And that's what I'm actually interested in studying. And Shikine is a great place to work at because a lot of our colleagues have done all the foundational work that's been really important for us to characterize the sites so that I can come in and do a lot of these next step work. So they've characterized the seep and found a good location for us to use that has about 900 PCO2, which is that end of the century level. And then we use the control reef right about 500 meters apart for our control sites. And what I wanted to look at was how are the fish found in these sites responding to this, the effects of the CO2 seep itself, the conditions inside. And for that, I looked at the neon damselfish, Homocentris coalesces. And if you look at this picture here, it's actually all those blue dots you see there are the neon damselfish. And you can see there's actually a lot of blue dots in all these photos. And there's actually one of the most abundant fishes found in both sites. So we know that these fish are here, and they seem to be doing well. And actually the fish that we collected from the seep in the summer were reproducing too. So they're actually reproducing within the CO2 seep itself. And what I was interested in is, well, it seems like these fish are doing great in these sites, but what are the mechanisms behind their adaptation to this site? So what we did was we looked at the transcriptomics of brain and gills of about a lot of fish around the island from the seep itself, which is in red here. And we were really fortunate to actually collect a lot of fish from control sites around the island to kind of get this overall trend. Because I wanted to look at this baseline trend, we wanted to compare how fish are from different control sites around the island to look at some fundamental genes that might be responsible for adaptation to these conditions here. And because Shikine Island, with the help of Sylvain and our colleagues at Shimoda University, it's actually really accessible for us. So we were actually able to sample multiple times within the year. So we did some sampling in the summer and the winter. And I'll kind of present some of the results that I have from this. So the first thing we needed to do for a lot of the genetic analysis is actually sequence the genome. For that, we went ahead and collected some fish and did a nanopore sequencing to get a reference genome for our molecular analysis. Here's just some stats on the genome that we assembled. It is a fragmented genome, but it's actually got 96.5% completeness for the Busco value, which is kind of a benchmark to show how complete this genome is. So we have a good reference for us to do a lot of the molecular work. And in brief, just the methods that, well, I'll be talking a lot about differential gene expression patterns today. And it's basically, we looked at we extracted RNA from the samples, sequenced it, and I compared how genes differed between the seep and control site. And I'll talk about genes being up or down regulating, meaning more copies abundant in the sites we're comparing. And I'm always comparing seep to a control site. So if I mentioned that some genes were up regulated, these are actually, in comparison to the control site, more of these genes were present in the seep population, the fish that we catch from the seep. And then if it's down regulated, it's vice versa. So the first thing we did in the summer when we went, we actually collected from 3 different control sites around the island. So you see that there's this green, magenta, and blue sites. And we compared the fish that we collected from there, from the CO2 seep in Shikine itself. And we looked at the brain and gill genes and which ones are differentially expressed. And if you look at the Venn diagrams here, this is just a number of differentially expressed genes. You do see that there's actually a lot of variability in terms of the genes that are differentially expressed across the different sites, which in differential gene expression analysis, it's actually really interesting because it's always relative to what you're comparing to. But if you look at multiple control sites, you actually see some overlapping genes that are differentially expressed, which kind of indicates that these genes are probably core fundamental genes that are involved in adaptation to OA at the CO2 seep. And I'll talk a little bit more about those genes in a bit. But when we went back in November, we actually did the same experiment, but collected from the seep and control site. So now I'm comparing seep and control from July and November. And there's actually some variability in the gene expression patterns again, but there are some overlapping genes also. And we'll talk about these genes. So you have seasonal variation in how these fish are responding to OA, but there's some core genes that seem to be consistently differentially expressed. And when you looked at all the seasons and tissues, we actually had one gene that overlapped that was consistently differentially expressed and upregulated. And these were genes involved with, this is endothelin, which is a gene involved in regulating blood pressure. And these trends in terms of gene expression patterns actually kind of make a lot of biological sense. So a lot of the genes that we saw were involved with blood oxygen regulation, which makes a lot of sense because one of the biggest challenges for fish in conditions of ocean acidification is adapting to that acidosis condition. So you need to regulate CO2 levels within the body. And we actually do see a lot of genes involved with that differentially expressed. And here's a heat map that's kind of summarizing the expression patterns across the two sites. So we have SEEP in red and control in blue with the different seasons and tissue types. And this is a Z-score, which looks at the expression patterns at comparison to control sites. So if you see warmer colors, it's upregulated genes. The cooler colors are downregulated genes. And we consistently saw genes involved with hypoxia response or regulation of oxygen differentially expressed. And in the gills, there were actually a lot of genes involved with vascular tissue development and also hypoxia response genes differentially expressed. Another gene that was really interesting that we found was that carbonic anhydrase was actually downregulated in the gills. And this is a gene involved in CO2 transport. And some really cool study in aquariums have actually shown that if you have a gradient of carbonic anhydrase, and so a reduction in carbonic anhydrase in the gill, and actually some in the body, it creates this Bohr root effect that kind of helps to pump O2 into the tissue and remove CO2. So this was actually happening in the gills. We actually found downregulation of this gene, which kind of shows this real life example of how they've demonstrated fish may be able to do this to help oxygen delivery in an experiment setting. So we saw some genes that actually mimic what we've done in laboratory settings, which is really interesting. In the brain, we actually saw a lot of these heat shock proteins, which are genes involved. They're great biomarkers for stress response, and they were all downregulated in the brain. And those were the genes that were over half of the genes that were overlapping between the two seasons were heat shock proteins in the brain. There are also many genes involved with immune responses and other genes such as circadian rhythm genes and stress response genes in the cytochrome P450 regions. These were all consistently differentially expressed across the different tissue types. And I've kind of shown you that, well, there's all these genes that they're using to adapt to the conditions of OA. But one question is, are they actually being selected for in the population of fish at the site itself? So this is where we go back and we looked at all the fish, and we actually collected some tissues from fish all around the island to look at the population structure of the fish found inside the seep and around the island. And what I did was I did some rad sequencing, which is just restriction site associated DNA sequencing, where you look at SNPs or single nucleotide polymorphisms within the population to see if there's any differences between. And with that, we had about 1.3 million loci with about 260,000 loci that had a SNP in it. And after filtering and looking for SNPs under selection, I removed the SNPs under selection and just looked at neutral loci. And we found that if you look at the if you kind of look at a principal component plot of just a discriminatory analysis, these actually, all the fish from different locations actually group out to their own location, which is really interesting. And you do see some very low FSD values. But I'm not saying that there's different populations, there's just some population structures that are present between the SEEP and other control sites. But one thing that was really interesting for me was that there's actually 32 private alleles that were unique to the SEEP population itself. And it's not just one SNP being present in the fish that are found in the SEEP itself. There's actually about 25% of the fish had 25% of those private alleles. So there seems to be maybe some environmental filtering or some assisted adaptation for those SNPs that are only present in the SEEP itself. And another thing that we wanted to look at was actually, well, there's a lot of loci that are under selection. And what are those loci that are under selection associated with? So I used two different programs to look for outlier loci, which are loci that are under selection. And I looked at which overlapped in between and just those genes that are associated with those loci. So these are some I had about 2,400 common outliers. I just mapped it back to the genome that we assembled and looked at the nearest gene associated with the common outliers. And we found out that those closest genes to it, a lot of them were actually associated with development. So either animal organ development, a lot of nervous system development, or axogenesis, development of organs, neurons, sorry. And we find that there was a lot of these developmental genes that seem to be under selection in the population within the seed. And this kind of comes down into this thing, because a lot of fish have these pelagic larval durations where they actually broadcast spawn, and larvae are in the pelagic water swimming across. So they should be able to swim all the way around the island and be present in any location, but there might be actually some selection within the seep itself to select for individuals based on development to let them settle back into the seep. This is something that we're looking into right now. We're doing some further analysis to look at what are these SNPs actually and what effects do they have. That's something that I'm working on right now to kind of look at, does this SNP actually cause a mutation that is going to have a difference? And that's something that we're working on right now. But it could be that Shikine might be filtering for species that are more tolerant of OA conditions, which could adapt to future conditions of OA. So this kind of summarizes what we've looked at the CO2 seeps. And what can CO2 seeps actually tell us about responses in fish to OA? So these COT seeps, as Sylvain mentioned, are great natural laboratories for us to study the effects of OA. And by looking at multiple control sites across different sites, and then the ICONA goal is actually to look at multiple ICONA sites. So you actually might be able to find some foundational genes that are differentially expressed, that are genes involved in responding to ocean acidification. And then what we wanted to look at was, are these actually products of selection itself? And that's something that we're working on right now to kind of identify population structures, SNPs that are present in a population to see if those genes are actually being selected for. And in future work, we're also thinking about perhaps using some light traps and things like that to collect larvae that are settling into the seep to see if you see any genetic signatures there. And I think Marco is going to talk more about why these sites are so important, but these sites could potentially be sites that are giving fish a head start to adapt to future ocean conditions. And with that, I'd like to thank everybody who's helped with this project. This is a very expensive project and a lot of sequencing was done and things like that. So none of that could have been possible without all the funding, with ICONA flying us out to all these places to have the opportunity to collect them. I've been really grateful. And all our collaborators that have been truly helpful and great people to work with. And with that, I'd like to thank everyone.
Thank you, Michael. Very nice to see more and more. We start to see patterns, you know, in chords. Carlos in Boracay showed the environmental filtering, selection of alleles and stuff. So we kind of start to see patterns, and I think this is very much why these sites are very important, and how maybe they can help us in finding ways to adapt to high CO2, or at least to help marine ecosystems maybe adapt to high CO2. We have some time for some questions. So please raise your hand or write it in the Q&A or in the chat. I'm not really sure actually, but feel free to ask questions to Michael. Of course, we'll keep some time at the end also, so that we can have a longer discussion at the end. Okay, random question on fish. This is interesting actually, but it's very important for industries, I think. How does OA, well actually, how does OA affect the taste of fish? Do you mean the how fish taste, or can they feel taste? But I think the taste of fish, do they taste better or smaller? Well, you fished in Shikine, so but I don't.
Definitely.
Yeah.
But in hindsight, if you think about like farm-raised fish, they actually are subject to higher conditions of PCO2 that are super extreme, like 10,000. So some people do have a preference for wild-caught fish versus farm raised. I don't know if the taste, I don't know if anyone's actually looked at the taste itself, but I know in Shikina Island, the fishermen don't collect fish inside the seep. And it's because they say that they have these, they do seem to be a little bit more darker in color. We don't know if it's because the habitat's more turf algae and that's causing it, but in Japan, it's, you know, aesthetics is a big part of, you know, the value of these commercial fish. So no one's done an actual taste test itself. I've ate the fish in there and I think they're just as good. They're pretty good. I mean, you know, we had a good time barbecuing up some of the bigger fish that we've caught, but we do see some darker colored fish, and that was actually something I've thought about, was that, you know, they seem to adapt to the surroundings with that more turfy dark colors, and that's affecting at least how fishermen perceive the seep. No one really goes in there to collect anything because they're like, oh, it's, you know, it's not a clean area, is their perspective on it, which makes it nice for us to study the seep because they're not taking the fish from there. But I don't know about the taste itself, but I thought they tasted fine.
I agree, but you're making a very, very interesting remark for like aquaculture, where they actually, when they farm fish, it's in an actual like very high CO2 environment, and it will be maybe like the alleles that you found, you know, they could be good to get a more like high production in aquaculture too, you know. And then like for the aesthetics and stuff, like actually we had a small paper on the ecosystem service for tourism, for example, and as you said, like it's very turfy, you know, like less bright colors fish and stuff, and the people are maybe less interested also in looking at these ecosystems. Okay, we have another question from Murat Belvemis. Sorry if I mistake your name. Very interesting talk. Are there significant difference among transcriptome of fish in control sites, such as difference in their SNP? So are there significant difference among transcriptome of fish in control sites?
Like comparison of control to control, maybe, is that?
Maybe it's the question, yeah.
Because, and this is the thing with a natural system and transcriptomics in general, is we're just looking at differences. It's a relative difference to A and B. And that's why I looked at multiple control sites, because there's lots of factors that can play into this. You know, in a worst case scenario, it could be when I'm transporting fish in a bucket to the lab, they sat there too long and there's affecting some effect that I don't know, or the sites that we're looking at, the control that the other control sites, I know you've characterized them all, but the other control sites might actually have something else leaching in. There could be some runoff, things like that. So it's nice to have multiple control sites to look at that against the seep. You will always find differentially expressed genes, even between two control sites. You know, two aquariums next to each other will have some differences, but that's why I think using multiple control sites, multiple seasons, you get more of a robust result. And for the SNPs, we didn't find any private alleles, so this is more of like a theoretical thing, but you looked at all the different 1 million sites that we looked at, and the chances of just the fish in the seep having these SNPs and not everybody else is actually statistically really improbable just by chance. So that was something that we're actually looking into more. And I need to still work on this, which it was my PhD project, but I'm working hard on it to get it done before.
Yeah, thank you. Yeah, I agree. Like multiple control is the best approach, and repeating cross-season the best approach is often very logistically and also financially difficult, but if it's possible, it's the best. And here again, I hope the ICONA network can help. Okay, thank you very much, Michael. We'll move to our next and last speaker. Oh, we're mostly in time. Marco, if you're here, I think you had a problem with camera. So
I
yes.
still have a problem with the camera, but I can hopefully share my screen. Let me know if you.
We can see, but we can see the PowerPoint, not the presentation.
Yeah. Is it now okay?
Yes, it's now okay. So Marco Milazzo from the professor at the University of Palermo will be talking about threats and conservation value of shallow hydrosome event. I think after these two talks, you can see the potential If you want to know more, also look at the previous OA week. There's lots of results that have been presented. And Marco, now it's up to you to convince everyone that these systems are super important.
Yeah, thank you, Sylvain. I'll start with a slide of yours. Actually, you just presented. This is the risk of working together for so long. So I stole it. And this slide basically says that shallow hydrothermal vents can be considered for sure natural analogues if you look at ocean acidification, but also natural laboratories if we enlarge the scope of our studies. And I won't spend more time on this, but we recently ran a short review on the in-field studies on vent around the world. And these results basically show that in many SIPs, one of the most common approach for ecological studies, specifically community ecology, was to use the gradient from extreme conditions, which are on the left, the main venting area, which is often extreme. I will talk about this later on. And then a gradient towards ambient conditions. In between those conditions, usually you find projected sites, which mean sites in which pH and CO2 concentration can be the one forecasted by the end of this century. So if we look at different studies, ecophysiological studies have often a similar approach. So they look at the whole gradient and see differences in organism and species, community shifts in the case of the ecological studies. But if we go on, then we have microbiologists that often give a focus on most extreme conditions. And there is a reason about this. And from time to time, compare those results with controls. Of course, this scheme is just a general scheme, and there are many exceptions about this. And then geochemists are mostly focused on the more extreme sites, looking at the bubbling and chimneys and the most active, geological active area of each shallow hydrothermal vents. And then we have genetics. We have had some examples just before me, and the idea of genetics is hopefully to look at potential adaptations in those conditions on many different aspects and processes involving transcriptomics and other responses. And these approaches compare the vent site with a different CO2 level with external controls, multiple controls, as just Michael said. But looking at this, given that my background was also marine conservation, I started thinking about the conservation of value of these systems. And in the literature, but also if you do interviews to the public and even some scientists, shallow water vents and seeps are generally considered not to harbor a high abundance of resident macrofauna or high level of endemism, so species which are typical of such environments. They are not to be threatened by human activities. They are not considered to be threatened by human activities. And for most of the studies I found in the literature, they do not have the same conservation value as other shallow marine ecosystems. So therefore, even regarding shallow marine vents, whether conservation should be considered is still a matter of debate. For sure, there is more in the literature by now, paradoxically, about deep vents, and this is because these environments, which mean well below 200 meters depth, are threatened by different human activities and by biotech harvesting, seabed mining, and other high impacting activities, and host very peculiar and endemic species around the world. So if we want to tackle the conservation of such systems in shallow waters, I think we should not only focus on ocean acidification studies that look at the gradients, but we should involve also the more extreme conditions, which are for sure of great geological and biological interest. So the presentation outline is, first of all, to put shallow water vents and seeps on a global map, to show their role in providing critical functions and in sustaining surrounding ecosystems. This will be done through some examples, the presentation of some examples recently published in the literature. Then to evaluate the exposure of these systems to 3 direct human non-climatic pressures like land-based human impacts, bottom trawling, fishery, and seabed mining. And finally, to evaluate actual conservation schemes of these neglected ecosystems at global scale. Before starting with some details, we should make a short presentation of these systems. On the left, you can see many diffuse venting field, bubbling field. They are both rocky, in some cases can also be on soft bottoms, on sandy bottoms. And the characteristic of shallow water hydrothermal vents is they are present up to 200 meters depth in what is called the sunlit zone. So basically you have light that can access till this depth. And they are mostly and more often restricted to volcanic active regions of the planet. They mainly originate from the interaction of seawater and meteoric fresh water with magmatic volatiles, primarily water, carbon dioxide, SO2, H2S, and these volatiles usually crossing the rocks and the minerals underneath the seascape, lose alkalis and take up elements like silica, iron, and they are enriching those in those elements. The characteristic of the fluids and volatiles emitted by these systems is that we can have both cold and hot up to more than 200 degrees Celsius, and pH ranges between 3 and 10. And the most common, the way they appear to us, is diffuse bubbling vent fields or chimneys, like the example we see below here. And another important characteristic of this system is, like their deeper counterparts, that they do show steep physical chemical gradients, and they are rich in valuable minerals and metal-rich deposits of great commercial interest, like their deep veins, like gold, copper, iron, and so on. This is why in the last few years, there is an alarming evidence that subsea mining can move closer to the shore in the continental shelf, so within the first 200 meters. And this is mainly due to the great technology and challenges to reach the depth of the ocean. And those places, those shallow places, could be much easier to be reached and exploited for this reason. And this has also raised many concerns, and you can see a recent paper published on the threat of shallow underwater mining of those systems. The way they affect shallow vents is really heavy. They generate plumes and sediments that can affect the biogeochemistry of the seafloor for up to a few kilometers away from the mining site, and for decades after the seabed mining activity occurs. So to put the shallow water vents on a map, we used some public databases and some databases we built up through a literature review. And I will present only the data on hydrothermal vents field and volcanic hydrothermal vents, but the analysis were also done for mud volcanoes and pockmarks, which often emit methane, but I will focus my results on shallow vents. In each of these databases, we collected the geographical coordinates, so point locations, depth, and type of vents or seep present in these these databases. We then cross-checked the activity of these vents going on the literature, and we considered them only if there was a study in the last 5 years. And this is how they are located. Of course, we are looking only on those occurring shallow, so up to 200 meters depth. And above you can see some details. Just to understand, hydrothermal vents are the triangle, mud volcanoes are the quadrats, and the pockmarks are the pentagons. And volcanic hydrothermal vents, either in the form of smoking chimneys or diffuse bubble fields, were the most recorded vent and seep type, and represented the 64% of the vents recorded in those databases. And let's go now to understand better the diverse geochemical patterns and processes occurring in these systems. First of all, they emit nutrients, gases, of course, and other reduced compounds. And since they occur in the sunlit zone, they can take energy from 2 primary processes, photosynthesis and chemiosynthesis. This is something very peculiar. Of course, the deep sea vents do not have the photosynthesis, and these plumes can go expand and interest many kilometers away from the vent site. And so you can have a great nutrient export in surrounding ecosystems. And of course, also these processes like photosynthesis and chemosynthesis is supported by an extreme high microbial taxonomic and functional diversity. This is the first example recently published on Science, suggesting the iron fertilization from a shallow active vent in the Tonga volcanic arc, which was recorded in a very oligotrophic, nutrient-poor region of the Southern Pacific. But according to the plume enriched in iron, which is a limited element in this region, these researchers recorded an onset primary production up to some hundreds kilometers away from the vent sites, and also hypothesized a regional CO2 sink into the deep ocean, because this element stimulates the nitrogen fixation of some planktonic cyanobacteria that sequester CO2. Another example showing the importance and strictly related to the high microbial diversity and both functional and taxonomic is a recent review which was published suggesting the key role of microbiota also in shallow vents and seep as ecosystem and engineers and habitat formers. Specifically, I will go quickly. 5 main habitats were established of microbial origin were established in general from microbes. And 4 of them are present in shallow vents, like microbial mats, microbial forest, microbial mineralized habitats like chimneys, and microbial outcrops. This is quite important because this kind of role from microbial communities can also support many associated species in this system. I will present briefly an example here in Sicily, in Panarea Island, in which we are now studying shallow chimneys, iron-rich chimneys at 70 meters depth in a place called Smoking Land, where more than 200 chimneys up to 7 meters high are present. And this is the detail of the place. In the last 2 years, PhD student of my group, I will present you just later, is collecting many data with many different techniques, and the combination of techniques is directed towards the assessment of fish assemblages associated to these systems. So she looked at different habitats like chimneys, rocky control, which means there are no potential influence of vents in the area, rocky Panarea, which could be affected by vent activity in some way, and sediment control, sediment Panarea in the same way, and vent sediment in Vulcano Island. I will present data only from BRAV, this technique here below, which is a baited stereo camera to assess fish assemblages, and those samplings occurred at depth below 50 meters. These are the results. As you can see on the left, chimneys in red host a higher abundance, and this abundance is comparable with other rocky areas, so other ecosystems, both in the area, so under the potential influence of any potential events in the area, but also quite far away from this area. While looking at the species richness, it show a lower abundance of species, a lower number of species, but species composition is quite comparable with rocky controlled rocky areas, which means that communities do have a high similarity with other habitats of conservation concern, like rocky reef in the Mediterranean. While the sediments, again, the vent sites host even comparable species richness and a higher abundance of the other sediments considering this study. Julia has also worked on some species and has also recently showed that many important mesopredators, like some important rays, do prefer areas closer to this chimney field. And the reason is that they for sure find more prey in this area. Another example is the importance of this system as genetic reservoirs. And this study was conducted, was carried out in Baia di Levante in Vulcano Island, so in the Aeolian archipelago, and we were able within the Two Frontiers project to isolate an incredible organism, a cyanobacteria, whose name is Cyanobacterium eponymum variety vulcano, which has a very fast growth rate, doubling its biomass every 2 hours, a faster sinking than any other known cyanobacteria, and a maximum growth and photosynthesis at high temperature, 45 degrees Celsius. This for sure has a strong potential to be applied for mitigating climate change via natural carbon sequestration of CO2 in the atmosphere. So after the isolation, now we are in the phase of experimentally, and this is an example on how this system could provide us nature-based solutions with a great potential for mitigating also this impact. And assessing, now I will briefly speak and run, because I think we are running out of time, about the 3 main human anthropogenic pressures that we consider: land-based human impacts, seabed mining, and bottom troll fishing. They basically occur in these systems because they are quite shallow and often close to the coast. So coastal urbanization, ports, mining on land, and also agriculture, they are all potential drivers of change from terrestrial inputs to this system, but also a physical impact by bottom trawling and, of course, seabed mining. We used a human modification index from NASA. This index basically considers the combination of 13 different land-based anthropogenic activities and it is available on a global scale with a very good resolution of 1 kilometer. We filtered the coastal areas and considered a range of 0, no human modification, to 1 complete human modification, and assess how these sites overlay with and the associated land-based activity with shallow vents. In this case, you should look at colors. The worst is the red, the best is the green. And as you can see, most of the red dots are located in the Mediterranean Sea, but also some of them in Japan and in Southeast Asia. And shallow hydrothermal vents, mostly located in those areas, exerted a high overlay with those activities. We then consider trawling. As you may know, with a delay of just 2 days, you can get information on trawling fishing effort all over the world, and there is a database that is provided by Global Fishing Watch for the transparency of ocean activities. And we overlaid the data of the apparent trawling fishing effort as hours fishing at a distance less than 1 kilometer over each site we consider in this study. In this case, you should look at the sides. The shape means the different seep type. The sides mean the higher the sides, the higher the hours of fishing this system. And then we assess the overlap with potential mining, and particularly on massive sulfide deposits occurring in shallow water, so the yellow dots you see there. But from this, we filtered the one occurring above 200 meters depth. In this case, you should look we assessed this as a distance from the vent and seep site, and we reported only on a global scale the MS as massive sulfide, which are those habitat quite rich in mineral resources and metal-rich elements. Finally, we assessed the spatial overlay of those areas with protected areas, existing protected areas, considering different levels of protection: highly protected, partially protected, less protected, and unprotected. And we considered the MPA name, MPA type, and the different PA protection levels from 2 global database that reported here. And these are the results. I will go straight to the main results. Only, let's say that 95% of shallow water hydrothermal vents have no protection at all from mining and trawling. Most of them, particularly in the region of the Mediterranean, have a potential direct impact with human land-based activities, but also in PNG with mining on land as an example. But distribution of protection around the planet is not constant, and the higher protection we can see is basically in Northwest Pacific, and specifically in the Mariana Trench, which is under the US exclusive economic zone. And this is concerning because the US has created very large marine protected areas to meet the 30% of protection of their EEZ waters, but focused this protection in overseas territories. And this is raising concern on urbanization of coastal areas in the continental US in the main country. And another important result is that 16 of 22 biogeographical provinces hosting shallow vents do not have any form of strict of protections, and this is quite concerning from an ecological and biological point of view, as this is unrepresentative. I won't spend any word on the cross between the different impacts, but there are some interesting results, and I go straight to the concluding remark. In this presentation, we hopefully demonstrated that these systems may provide important nature contribution to people, and they are quite threatened by many human activities and are largely underrepresented in existing conservation schemes. And there are regions in the world, like the cold and warm temperature ocean, that have no highly protected areas at all, while shallow vents in distant and remote islands at the highest level of protection, which could be good, but is for sure is not enough. Conservation planning approaches and environmental impact assessment should be extended by soon to these ecosystems, given their ecological relevance. And for sure, a shift is needed in how these systems are viewed by scientists, policymakers, and policymakers. I wish to thank finally ICONA and the group, the Italian, Japanese, and French New Caledonian group, the Two Frontiers project, with whom we now are working on basically on microbial functions within this system all over the world. Of course, my group at the University of Palermo, and thank you also to the funders and to you for your attention. Sorry.
Thank you, Marco. It was a great talk. I think first, like the cyanobacteria story in Vulcano was presented last year at the OA week, so you can go to YouTube to watch it. And there are even today, some more talks showing how relevant these ecosystems are to understand future ocean, but also to find solutions. And it's very sad to see them neglected from conservation. So we are already over time. I will continue until the secretariat of GOA-ON tells me to stop, but okay, 5 minutes max. So we have 5 minutes max, and if you have a few questions for Marco, but also for the other, we can jump back to the other talk anyway. So as always, please raise your hand or write in the Q&A or in the chat. One more time, also a reminder that if you know some of these systems, that have been yet explored or that you're working on, please reach out. It would be great if we can collaborate and if we can help in any way. We did lots of not mistake, but when we started using these systems, and we will be happy to share our failure and success with you. I just got 5 more extra minutes, so if you have any questions, now is the time. Otherwise, I think we'll have to close the session. Rina, you want to close the session? I can say it.
I think everybody's just been overwhelmed with the amount of information.
Oh, okay.
There's one question.
Okay, it will be our.
If you want to answer that.
Yeah, it will be
That one.
Our last question then. It's a nice question. What are the ICONA next steps to push for the conservation of the vents? Margot, it's a question for you. But if any speaker wants to jump in, feel free.
Yeah, for sure, deepening potential solutions belonging to these systems, and specifically looking at climate change scenarios, because we can have in some of these systems higher temperatures and of course higher CO2, and this could explain the presence of genetic reservoirs like the strain of cyanobacteria with very important characteristics. Then, and the other is to expand the number of studies to many unknown areas in the planet, and that's it.
Yeah, I agree. I think like we need to very much like push show the potential and keep showing the potential of these sites and other unknown sites, and and yet some people in Taiwan, I know the people in Taiwan actually in Turtle Island, and yes, some endemic crabs I know of, but yeah, there's lots of organisms that we still need to understand how to survive, and that can teach us a lot about it. But sorry, we have to close this session. We're already late. Thank you all for joining, and thank you, GOA-ON, for these wonderful opportunities. Thanks to all the speakers. And Katerina, if you have closing remarks to do.
Brilliant. Thank you all so much. This was an amazing start to OA Week 2025. Thank you to the audience for being so engaged and asking so many questions. Thank you to our speakers and presenters. And please don't forget that you can consult the website on the QR code shared here for any other sessions on OA Week, and also to join and become a member of GOA-ON, so that you will always be among the first to be informed of anything that's happening in the OA world. And with that, thank you all so much.
Thank you.
Thank you very much.
Thank you.
Thank you all. Bye. Bye.
Bye bye.